Professor and Sanoji Samarakkody from Quinnipiac University, USA, a Sri Lankan female scientist who has excelled in her career in chemical sciences at an international level. This is the first webinar under the quarterly webinar series. The objective of this webinar series is to share the experience and knowledge of highly achieved professionals in the field of chemical sciences to inspire future generations in pursuing their careers in chemistry. So this webinar is under the theme where the webinar is conducted by a professional in chemistry who is currently serving at international level. With that, I would like to welcome you all again, and over to you, Dr. Amba.
Dr. Ann Sanoji Samarakkody completed her graduateship in chemistry program at the Institute of Chemistry, Ceylon in 2009. For her undergraduate research, she studied secondary metabolites under the supervision of Professor Priyani Paranagam. Then she served as a postgraduate volunteer researcher at the Department of Chemistry, University of Peradeniya, where she conducted research on liposomal drug delivery under the supervision of Professor Chandani Pereira and Professor Nedra Karunara. In 2017, Dr. Samarakkody earned her PhD in anatomy and cell biology from the University of North Dakota. Her doctoral research focused on the molecular mechanisms involving RNA polymerase II pausing during gene activation under the supervision of Professor Sargeni Shea. From 2017 to 2022, she was a postdoctoral researcher at the Department of Pediatric Hematology Oncology at the Boston Children's Hospital and Dana-Farber Cancer Institute, Harvard Medical School. Under the supervision of Professor Alan Cantor, she investigated the pharmacological enhancement of residual wild-type RUNX1 and FPD AML disease backgrounds. Dr. Samarakkody also served as an adjunct professor at MCPHS University, Boston, where she instructed laboratory classes in anatomy and physiology, and as a visiting assistant professor of biomedical sciences at Quinnipiac University. Currently, she is an assistant professor of biomedical sciences at Quinnipiac University. Her research focuses on understanding the molecular and cellular mechanisms that maintain cellular integrity. She is particularly interested in identifying molecular targets with therapeutic potential using cancer cell culture models and DNA damage inducing agents. Her lab investigates how small molecules can reduce DNA damage burden and reduce cancer predisposition. Additionally, her research explores the role of protein targets and small molecules in resolving the DNA damage response. So, Dr. Samarakkody, thank you for being with us today. Now I invite you to conduct the webinar.
Thank you very much, Dr. Samira Gunatilakar and also Dr. Dinusha Udukala for the introduction and also inviting me here today. I hope you all can hear me well. Again, I'm very excited to be part of this event today, and I'm going to talk about my journey from starting as a chemist and moving to become a cancer biologist. And during my talk, I'll bring up my research as well. And you'll notice a common theme where I do talk about small molecules and using them to face challenges, specifically in the context of cancer. So a little bit of an overview of what I'll be talking today. I will start off with my experience at College of Chemical Sciences, Institute of Chemistry, and then talk a little bit about my research in my PhD thesis project, and then also talk about my postdoctoral work looking into small molecule inhibitors, specifically in the context of cancer. And then the focus of my current lab, where I'm exploring small molecule inhibitors as a tool to reduce DNA damage burden. To start off with, I did my undergraduate work at College of Chemical Sciences. I was lucky enough to receive a scholarship through the Royal Australian Chemistry Institute to support my education at College of Chemical Sciences. And this is where I got my first experience in research under an incredible mentor, Professor Priyani Paranagamar. And I was able to conduct my research at University of California looking at antibacterial activity of secondary metabolites in endolyconic fungi. And this is where I decided I really liked research. I did work with my mentor for over 2 years, even a little bit after graduation, to wrap up the project. And I decided that the route of a PhD is where I want to go. And I knew I had some experience in working with secondary metabolites, but I wanted to expand my research experience. And a little bit of background. I was a student at Institute of Chemistry, as I mentioned earlier, but I was in a program where I was able to do my first 2 years in Kandy with professors from University of Peradeniya, and my last 2 years in Colombo. So because of that, I was familiar with faculty members in Faculty of Sciences. And under the mentorship of Professor Chandini Pereira and Professor Nidra Karnaratne, I worked on a research project targeting drug delivery through liposomal disintegration. Although this experience was short, it did give me valuable understanding and I was able to brush up on my critical thinking skills. And I really knew at that point, I really love research, and PhD is a route that I want to take. And unknown at the time, there was a common theme that I was drawn to, which was looking at small molecules, looking at drugs, and kind of heading in the direction of what I can do in the context of helping the healthcare research field currently. If I'm being completely honest, finding a PhD program is stressful, but it is a very exciting journey. I had a really strong foundation in chemistry through my undergraduate program, but I wanted to expand the horizons a little bit. Knowing the amount of chemistry and the biochemistry that I did at the time, I want to explore what really happens in the cell. And with that, I knew I wanted to do a PhD in the field of either cell biology or molecular biology. And I decided to apply to PhD programs in the United States. And this was mainly because it is one of the countries that allows PhD students to take classes. And since I did not have a strong background in cell biology, I knew if I wanted to be successful in my field, PhD programs in the United States would offer me the best chance of success. Again, we always talk about successful experiences, but I also want to mention not every application cycle might be successful. And my first round of applications was not successful. I realized that there were too many moving parts with the GRE scores not reaching the programs. And with all of that, I was not successful the first time around, but the second time I was successful. I decided to join University of North Dakota for my PhD, and I got accepted at the Department of Biomedical Sciences. And I'm proud to mention that I was the first Sri Lankan graduate student in that department. And I was drawn to this program because I knew I'm able to learn more about aspects of cell biology, molecular biology, and also human anatomy, and some of the fields that I was lacking. And biomedical sciences would give me that holistic view to where I want to go in my future direction. And my thesis work looked specifically at transcriptional regulation under the mentorship of Dr. Sergei Nechayev. I looked at a concept known as RNA polymerase II pausing. So during our biochemistry, we learn about initiation, elongation, and termination of transcription. However, in eukaryotic organisms, what happens is polymerase II does get loaded onto the promoter regions, and it would initiate transcription. But what happens in eukaryotes specifically is it would transcribe about 35 to 50 or so nucleotides from the transcription start site, and it would remain paused. And this is the concept that is known as RNA polymerase II pausing. And there are a lot of cues. So this image is to summarize a lot of things that are happening at the polymerase II pausing site. This is an important regulator of transcription. One might think by pausing, it is simply halting transcription, it is hindering transcription. But what happens is this enzyme complex is transcriptionally engaged. So it is waiting for an additional cues, could be internal or external cues, to continue with transcription. So this is a decision-making point in transcription. And as we all know, genes are in our DNA, but they need to be transcribed and translated. So this is one of the first regulatory steps in gene expression. So at this point, it could either undergo premature termination, where the complex gets dissociated from the DNA, or it can go into productive elongation, making the actual RNA that the cell can use to make a protein from. So very simply, we were able to show that RNA polymerase II pausing is retained, and it can also be acquired during gene activation. And what I'm showing you here is mRNA fold change with on a specific protein known as heat shock protein, which is the black bar that you see here. And based on the RNA levels, you could see that with each time point, the level of RNA is increasing, and we are also showing that the level of RNA polymerase II pausing also increases intermittently as well. So that was the first time we showed that genes can acquire pausing, or the levels of pausing can be retained, or even in certain cases, the level can increase in response to gene activation, really validating that it's the bottleneck step in transcription regulation. And after the PhD program, that was another decision-making point in my career, where I had to decide, where do I want to go from here on? And having the education I've had and the direction that I would like to take for my future, I decided against going into industry. I decided I want to go on an academic path. And I also realized doing a postdoctoral fellowship is really going to help me. No matter if I go into industry or not, a postdoctoral fellowship is always a wonderful thing to have, where you can brush up on the mentorship skills and the independent research aspect as well. And I decided to pursue a postdoctoral fellowship, not in industry, but in academia. And knowing all the teeny tiny mechanistic details that I knew about transcription, I wanted to then apply that knowledge and the chemistry knowledge that I had into more of a disease background. And I knew I wanted to work in a lab that was either associated with a medical school or a hospital, where I could do more of translational research work. And I applied to a few different places. And again, I tried to gear my search towards places that would allow me to do a little bit of translational type research. And I decided I want to be in the field of oncology. And again, like as I mentioned earlier, knowing what is happening at the level of transcription, what can I do next with the knowledge I had? How can I apply this knowledge into an actual disease background? And I was lucky enough to work in the Cancer and Blood Disorders Center at Dana-Farber and Boston Children's Hospital and Harvard Medical School. This is in Boston, Massachusetts. And this was a really fantastic environment for me, because I was able to learn a little bit more about cancer. And it's a great environment to be as a trainee, to increase the network of scientists that you know and you're involved with. Great place to build collaboration, great place to build a lot of the skills that you want as a scientist. The disease specifically, I did mention that this is a cancer center, and the disease specifically was a type of leukemia that is known as familial platelet disorder with a propensity to develop acute myeloid leukemia. So it is a mouthful. It is also abbreviated as FPD-AML. And this is what the lab primarily focused on. And this specific cancer is characterized by dysregulation in platelets and also in a specific cell type known as megakaryocytes. So these are the cells that shed off platelets, they produce platelets. And these cells are present in circulation, but they're primarily present in the bone marrow. These two images are showing the specific dysregulation I mentioned. So the first to the left, these are electron micrographs of platelets. And the first two panels are affected individuals, and this is in comparison to the healthy control. And what you can see clearly is the affected individuals have platelets that have abnormal shape, and they look a little atypical. And this is one of the phenotypes that the patients do display. So sometimes, even if patients have the platelet numbers in the normal range, they don't function as well. And because of these patients tend to have bleeding disorders, they bruise pretty easily. And that is one of the things that patients are diagnosed with in the clinic. And I also mentioned that these patients have atypical megakaryocytes, the cells that produce platelets. Healthy megakaryocytes tend to be quite large in size, and the affected individuals have megakaryocytes that are not quite as large, implying that these cells are not as matured. And another characteristic is these cells, the healthy cells, tend to have lobulated nuclei. So if you look at this stained picture showing healthy controls, you could notice that the nuclei that are stained in dark purple tend to have a more lobulated nature. However, the affected individuals have more like a fine circle of the nucleus. This indicates that these megakaryocytes are not as matured, and therefore they might not work as well. Another characteristic is, unfortunately, individuals who are diagnosed with FPD-AML have an increased risk of developing leukemia, which truly is an unfortunate circumstance. The reason is these individuals would have easy bruising. They would go into the clinic and get diagnosed with FPD-AML. And it's the reason why I said it's unfortunate is because these individuals they know that they might develop leukemia at some point in life. They just do not know when. So it is a really unfortunate position to be in. And the root cause of this disease is a mutation on a transcription factor known as RUNX1, also known as RUNX1. So this is a germline mutation, meaning it is hereditary, and it's a heterozygous mutation, meaning there is a mutated allele and then there is a wild type allele. Remember, all humans have two alleles for each gene, and these mutations tend to be heterozygous mutations. And knowing that there is a wild type allele in these patients, the project in my postdoctoral work tried to focus on, can we leverage the wild type allele using small molecules to increase the activity of the wild type so that it can surpass the dysregulation that the mutant protein is causing. So a little bit more about this transcription factor. As many transcription factors, factor proteins, it also has, it's a multi-domain transcription factor. It has a few different domains. And one of it is the DNA binding domain. And there are a few different regulatory domains that are shown in red and blue over here on this schematic. What is important is within these within these regulatory domains, there are tyrosine residues that do get phosphorylated. And this is a very dynamic phosphorylation and a dephosphorylation. And once phosphorylated, the transcription factor is inactive. And when it is dephosphorylated, the transcription factor tends to be a little bit more active. The nice thing is there is a group of enzymes that causes phosphorylation on this transcription factor. And there are a known class of inhibitors that act against this class of enzyme. So bottom line, we can use these inhibitors to modulate the activity of this transcription factor. And knowing that, we switched to a high throughput small molecule screen at the Harvard Medical School. And I'm just showing a screenshot of some of the small molecule libraries that were available for us to use. And we used quite a few of these libraries for our preliminary screens. And what I want to point out is the field is shifting towards small molecules that are already FDA approved. So if you look at these libraries, the dark blue bars are the small molecules that are already FDA approved. And the advantage of using FDA approved molecules is that there will be some preclinical or even clinical data that is available with these molecules. So that's what the field is trying to shift towards, just to ensure that drug discovery is happening at a timely and a not so delayed manner. And these are some of the data from the assays that I did through this high throughput screening. And what I want to point out is I understand there are a lot of molecules to look at. However, we did find out molecules that are targeting SARC family kinases and also CDKs, which are cyclin-dependent kinases. Both of these classes are known to regulate RUNX1 activity, and we were quite impressed to find out that without any filtration of the hits, we are picking up quite a number of SARC family kinase inhibitors and CDK inhibitors. And the ones with asterisks were already FDA-approved drugs, so we switched our focus towards these FDA-approved drugs. So having this preliminary data, we had to validate this data. And we did it in a few different ways. We used induced pluripotent cells from patients who do have this disease, and we tried to make hematopoietic cells or blood cells and the megakaryocytic cells using the induced pluripotent cells from the patients. And we tried to find out if we can, number one, address the DNA damage burden that these patients have, which is one of the main reasons why they develop cancer, because they're acquiring mutations. And with acquiring mutations, it increases the chance of developing cancer later on. So we first wanted to see if any of these small molecules can decrease the DNA damage burden. And I'm showing just one piece of data here, and we were able to show that SARC family kinase inhibitors are able to decrease the DNA damage burden, which means the chance of acquiring long-term mutations is low. And hopefully that would alleviate the phenotype of individuals developing leukemia later in life. And the next thing was looking at hematopoietic progenitor cells and also looking at the megakaryocyte, the large cells that I was looking at. And in comparison to the control, which is the DMSO control, we were able to show that with a couple of different molecules that we are able to rescue the number of megakaryocytes these cells are able to produce in cell culture models, again, using induced pluripotent cells. And I understand that I showed you only two pieces of data, but there are a lot more data, more than that. So with sufficient amount of supporting data, next phase, the findings from my project did move on to a clinical trial with NIH in Maryland. And currently there are 72 patients that are enrolled in this clinical trial. It is going to end next year. And the goal is to get enough phase 1 data to move on to phase 2 and 3. And one of one of the molecules was picked to move forward with in this clinical trial. And I am very optimistic and hopeful about the results from this clinical trial. So that brings to the end of the postdoctoral phase. And then I was at a point where now I had to decide what I'm going to do next. So a quick snapshot, I just want to show you that my undergraduate education was in chemistry. And then for a PhD, it was a cell biology PhD. And for my postdoctoral fellowship, it was a drug discovery project in a leukemic setting. So the reason why I want to show you this was because at each point in my career, it did look like I did kind of deviate from the traditional path. I did not go into a chemistry PhD, rather I changed my path, and then even at my postdoctoral, I changed my path yet again. And it could be daunting, but at the same time, if you approach it with a positive mindset, rather than thinking, is this going to be a challenge? What if you think, I am going to face this challenge? So I just want to share that experience, because I was a person who kept changing my career path at each point. And I also want to mention that it really depends on what you want to do with your life. And at each point, I decided in my life, I wanted to do something different. I want to do something more. And that was one of the main reasons I made these changes in my career. And then I had to decide, what am I going to do next? As an international graduate student and international postdoctoral fellow, one of the main challenges that I faced was acquiring funding, because there are limited opportunities, and I was not able to be extremely successful in acquiring funding as a graduate student or as a postdoctoral fellow. And with that, and the second thing was, I did work as an adjunct professor while I was a postdoctoral fellow in Boston. And at that point, I noticed I really do enjoy teaching. And that was the second reason. And I also realized I do not want to completely give up research. I still want to be involved in research, maybe not at the level of PhD, maybe at the level of master's. And with all that together, I decided that I want to join a primarily undergraduate teaching institution with the possibility of teaching master's students and doing research with master's students. And that led me to Quinnipiac University in Connecticut. And here, the amount of work that I do in the Department of Biomedical Sciences allowed me to teach both graduate and undergraduate level students. These are some of the classes I teach. I teach a class known as Research Methods that is focused on techniques in molecular biology. For graduate students, I also teach fundamentals of oncology for graduate students, and also a cancer class for undergraduate students. I also teach biochemistry and immunology as well. And I want to emphasize these are the things that I mastered while doing my PhD and also during my postdoctoral fellowship. And the work here allows me to maintain an active research lab, and I'm able to use the same model systems that I've used in the past. My lab is primarily using cell culture as a model system, and I'm able to maintain a research lab, mentors undergraduate and graduate students in their research projects. And some students are thesis students, some students are non-thesis students. So I get to advise a wide variety of different students with very different goals that they're trying to achieve in their master's program. I get to provide service to students in terms of mentorship and advising in their career goals. And I'm able to provide service to the university through different opportunities and different avenues that would allow me to do that, including working with International Student Center and also doing some outreach activities by reaching out to local high schools to bring them here to give students more opportunity into the field of biomedical sciences. Talking a little bit about my work here in the lab at Quinnipiac, I still want to remain in the field of cancer biology, and I shifted from leukemia to breast cancer. I had worked with breast cancer models in my PhD program, and the significance here is 1 in 8 women in the United States will be diagnosed with breast cancer at some point in their life. And a majority of individuals who do get diagnosed with breast cancer do not have a family history of having breast cancer. And the burden of cancer still remains really high worldwide, despite of all the new therapeutic interventions, which really emphasize that there has to be more research that need to be done in the field of cancer, in my case, in the field of breast cancer. And the mortality rate of breast cancer still remains pretty high. And among all types of breast cancer, among all types of cancers, breast cancer is one of the leading causes of death in women worldwide. And there is a growing wealth of evidence that also imply the exposure to various chemicals contributes significantly to incidence of cancer. And again, I want to tie that statement into the statistics that I showed you, where 85% of breast cancer patients do not have a family history. So it is not genetic and it is not hereditary, rather it is something that individuals acquire. Again, tying back to my previous work, one of the main reasons could be that we are getting exposed to a lot of different chemicals through our lifetime. And even the groundwater nowadays has some contaminants in them. And with increased burden of exposure to different chemicals, some of those could be DNA damage causing chemicals. And some of these chemicals get accumulated in the adipose tissue. Some of these chemicals could be disruptive to hormonal regulatory pathways. And all of these could contribute to increased chance of breast cancer in individuals and in the population today. And I also want to emphasize that breast cancer is also diagnosed in males as well, but it is more predominant in females due to the hormonal variation that women do face through their lifetime. So thinking about DNA damage, some of this could be from oxidative stress or cellular stress, and some of that could be due to carcinogens. So there are 2 starting points that one could look at DNA damage, and these could lead to DNA breaks. These could be single-strand breaks, these could be double-strand breaks, and depending on if it is single-strand or double-strand, there is a whole set of proteins that are present in eukaryotic cells that act as effector signals. So they act as DNA damage checkpoints. And then the cell makes a decision of either repairing this DNA damage, and that could lead to cellular homeostasis, or there could be not sufficient DNA damage repair, and that could lead the cell undergoing either apoptosis or cell cycle arrest. And if the DNA damage is not resolved in an efficient manner, it could also lead to increased rate of mutation. And a little bit more about DNA damage, what these proteins are. So this is a simplified version. There could be stress signals, as I mentioned earlier. Some of those are external, some of those are internal. Could be oxidative stress due to reactive oxygen species, could be carcinogens that we are getting exposed to. So there are sensors, and they can undergo modifications, and there can be regulators of those proteins, and there are a whole set of effector molecules. And then these effectors, depending on what effectors are activated, cells can then have a very specific response. So this response in a big picture could be DNA damage repair, could be cell cycle arrest, could be apoptosis or senescence. So this is what my lab is currently focusing on. And our goal is to dissect into these molecular pathways and try to see if we can look at small molecule inhibitors that are able to alleviate some of the DNA damage burden that we have, and then we are getting constantly exposed to. Again, I want to emphasize that I really do enjoy doing research, and the current position I'm in is allowing me to do research. And during my career, unknowingly, I have acquired the skill set that I need to be in the position that I'm in right now. And also the experience in teaching had also helped me to be successful in my current position. With that, I would like to thank SLAAS and Dr. Ranmal Gunatilakar, and also Dr. Dinusha Udukalar for having me here today. And I would like to take the time to thank all of my mentors and professors for their guidance through my career, and starting from my school back in Kandy and Institute of Chemistry and University of North Dakota, and also Harvard Medical School, and also my professors and my mentors. And I would also like to thank the funding sources, without which none of this work would be, I wouldn't be able to do any of this work that I presented today. With that, I thank you again for your attention, and I am happy to take any questions you have.
Okay. Thank you, Dr. Samarakkody, for your informative and inspiring webinar presentation. Your research work is truly impressive and has provided us with valuable insights. So I believe the speaker is now open to taking some questions from the audience, as she mentioned. Um, I have a question in the chat box. I will read it out. Hi, madam. I'm currently a A-level student. I'm studying from BioStream, Cambridge syllabus. My question is that I am really worried about the path I should choose. Like, other than medicine, it's really hard to find best degree to study on for my bachelor's. So, can I have any recommendation about what world needs in the future? What path will be really valuable to go on? Thank you. So, I think that's an interesting question from a level student.
Yeah, I'm pretty impressed that you're thinking about 5 to 10 years from now, and that is pretty incredible, Prabhasha. Yeah, thank you for your question. So I think you said of any path other than medicine. You would really have to think about what you would like to do. So I know I didn't talk too much about the phase of A-level, but I knew I wanted to do chemistry because I really loved chemistry as an A-level student, and had family members who were in higher education, and they were chemists as well, professors in chemistry. So I had a tendency towards chemistry, and I decided to pursue chemistry. But then at that point, I decided, well, I need a little bit more about, I want to know more about cell biology and molecular biology. So it really depends on what you want to do. And if you are thinking about the medical field in a broad sense, I think if you do any undergraduate degree in, could be biology or chemistry, could be a combination of those, and some programs do have biomedical science undergraduate programs. And I would think any of those programs would give you the tools and the knowledge you would need to make that next decision for your next phase. So it really boils down to what you really want to do in the future. Again, I do not know if that answer is sufficient, but I think any field in biology or chemistry or sciences in general would help you. And I also want to add, not every PhD program is, so going beyond the question that I was asked, not every PhD program requires you to do a master's. Most of the PhD programs would look at your research experience. And if you do have sufficient research experience as an undergraduate student or as a postgraduate student, they might see you as a good candidate for a PhD program. And then the next phase would be, how would you present yourself in the phone interview? Some programs do have phone interviews. So how do you want to talk about your research? And because there are more than what you can put on the paper that you can present about yourself during a phone interview. So that truly matters as well. I do not know, Prabhasha, if I answered your question sufficiently, but I'm happy to answer a follow-up question on that if needed.
Yeah, thank you. So there's another question. Dr. Samarakkody, if you don't mind me asking, what was your undergraduate GPA? That was the question. Also, did you have ever consider opportunities in the other field? And if so, what made you stay in this one?
Yeah, thank you for that question. Honestly, it's been so long and I do not remember my undergraduate GPA. But in College of Chemical Sciences, there's a GPA component, but then there's also the classes. So I had a second class upper. I don't know if that's helpful. That's the only part I remember. I do not remember my GPA. So I wasn't
Yeah,
I
let
wasn't
me.
at the top of my class, if that's helpful.
I think in your time, the classes were given based on averages and
Yes.
not really based on the GPA. So, according to the criteria, like your GPA is like, I mean, your class is second class upper division honors. So that is based on average.
Yes. Um, yeah, that's correct. And thank you for that. And I want to mention I wasn't at the top of my class. So I wasn't a first class. I was a second class upper. And did I consider opportunities in other fields? So I think if you look at my career path, I have always chosen a different field at each point. For example, I started off with chemistry, and then I went to more molecular biology, and then I went into cancer biology and decided this is my happy place. But other than that, so by other opportunities, if you are thinking about maybe industry rather than academia. I knew I did not want to be in industry, and that was because I truly liked academia. I liked the environment in the academia a lot more. That being said, I also want to emphasize that each company is different. If you look, I can briefly talk about the environment in the United States. Each company is different. Startups could be much more different than more established environments. And some industry do sponsor visas. They sponsor postdoctoral fellowships. So it is quite different from one place to another. But I knew I did not want to be in industry because I like the teaching aspect of academia. And I really want to be in an academic setting for my postdoctoral fellowship because I knew I would get a better exposure. Like I mentioned earlier, I want to be either in a medical school or affiliated to an hospital because of the amount of learning I expected from my fellowship. And I know that would have been limiting if I went into industry, but if somebody's thinking about industry opportunities, industry postdocs would be ideal for them. And then just talking about opportunities, the second one is picking a primary undergraduate institution. That was, as I mentioned earlier, a few different factors contributed towards that decision. The first being I wanted to teach, and then the second being knowing how the funding environment is, knowing how competitive it is. And I did a whole lot of research into what it takes to establish as a new PI in today's day and age, and just the demand of that and just the stress that it might involve. I decided against it. That being said, there are lots of other Sri Lankan faculty who have gone into R01 type research institutions that have PhD programs and postdoctoral fellowships. So this is a choice I made for myself, and I decided this is what I want out of my life. I want to do a bulk of teaching. I want to inspire students. I want to encourage them to go into the field of research, and that's why I'm here. That's why I am where I am today.
Yeah, there's
Yes,
another
that's why.
question from Satnara Fernando. The question is, for an undergraduate in chemistry hoping to turn into medicinal chemistry, do you think it's essential to pursue a PhD? Are there any other alternative career paths?
There are lots of career paths, and I did a presentation last week in our graduate program about career paths, so it's kind of fresh in my memory. So there are lots of career paths. I think you need a PhD if you want to either go into academia, if you want to go into industry. Some senior scientist position would require you to have a PhD. And then if you want to become an investigator in industry or in academia, you absolutely need a postdoctoral fellowship, at least within the environment that the United States currently have. But that being said, there are a bunch of other career paths, and some of these we don't even think about. One could be like way different that what you're probably thinking is intellectual property. And that's somebody with a science background with sufficient experience in the field of law. If one serves as an intern in a law firm, they could also work in intellectual property. And then there's also scientific writing. There's also teaching. There is also, just going back to scientific writing, in certain institutions, there are dedicated scientific writers for their outlets. So for example, a lot of medical schools and hospitals have their own newsletter, so to say. So they do have scientific writers that talk about the therapeutic options that the institution is doing or any research that the institution is doing, and kind of translating it in a way that a lay person could understand the research that is done and the significance of it. And then there's also another part of scientific writing where one could be at a research institution, and their role would be to support the investigators in their writing. So this could be either a literature review, this could be supporting information for a grant. So it tends to be a little bit more scientific, and that is another avenue that one could take. And in addition to that, there are a lot more. So if you think about medicine, there could be genetic counseling, there could be epidemiology, and then if you think about research, there could be a biomedical research scientist, microbiologist, a lab manager, assay developer, because a lot of research institutions tend to have what's known as research cores. So these core, a core could be dedicated to sequencing or microscopy and high throughput assays, as I mentioned earlier in my work. So there are a lot of core services that places offer. So one could go into that. And less common is industry sales. And there are product specialists, and then there are sales representatives that do have science backgrounds, depending on your education level. Sometimes there are master's level positions, but then if one has a PhD, they could be a scientist at an industry where you provide customer support specifically to researchers who need support with the products that the company is trying to sell. And then in addition, there are like assay developers in industry positions that are also involved in R&D as well. So there are a lot more that one could explore. Again, it comes down to what do you really like to do? What do you really want to do? That was a long answer.
Yeah, that's a good answer. That's an informative answer. So a couple more questions. As an undergraduate, I will read out all the three questions. So first question is, as an undergrad, do you encourage getting prior research experience, and how to? The second question is, could you please explain how does AI technology impacts on cancer biology?
Right. So I'll answer the first one. So if I would encourage anybody to do research in general, and I think I am also biased, and this is because if you don't try it, you don't know if you would like it. For example, I had a student 2 semesters ago. They didn't know that about too much about the PhD track, and they tried a thesis project and ended up really liking it, and now they are pursuing a PhD career, PhD route. So you wouldn't know if you would like it if you don't try it. But as I briefly mentioned during my talk, sometimes it's hard to get a paid position. Sometimes you would have to go for a volunteer positions, and I did that when I was in Sri Lanka because it again, I understand that it could be really competitive, it could be challenging, and sometimes volunteering is a way to go. And that is what I did. I wanted the experience, I wanted to learn, and I decided to be a voluntary researcher. So I would encourage anybody to do research, to acquire some research experience, no matter what it is. It might not be what you were looking for, but maybe you might end up liking it better. So I would personally, again, I love research, so I am biased, so I would encourage students to at least try research to see if you would like it, because the world needs a lot more researchers, and there's an increased need for researchers, and I would truly encourage. So to answer the second one, AI, artificial intelligence. So it is a helpful tool and also a challenge. And that is something that as educators that we are facing on a regular basis, because although it can be helpful for student learning, it can also hinder it in a way if a student is relying on AI too much. But in cancer treatment, so AI is not at the point where it is effectively used in cancer treatment strategies, but prediction models are used. So for example, so this goes into a little bit of computational chemistry and computational biology, not really AI, but kind of like a middle grounds to that. So the discovery of new targeted therapies, they do run through simulations and some AI. However, all of those targets need to be validated by an actual human. And thinking about streamlining those processes, although it is not technically AI, a lot of high throughput sequencing or high throughput facilities do use robotics to get the work streamlined and to increase the accuracy. So it's not technically AI, but that's where the field has advanced. So the contribution of AI, so there is a small part to it, but as of now, currently, the bulk of the work is done by actual scientists pipetting at a bench. So that might change within the next 5 years. However, I don't envision an environment where the human contribution is completely replaced. Maybe that was where your question was heading. And there are a few more questions.
Due to limited time, we will take only 2 more questions. So there are questions. Funding, a question about financial fundings from medical and educational side. Yeah, and then the other question is from Devinti Pereira. For a person who is looking to get a PhD and industry-related career path, is it best to go for a PhD straight after the bachelor's degree, or to gain some experience before the PhD? So if to do a PhD after some time after working in the industry, what are the good opportunities to look for? So that will help me to get a better chance for a PhD. And the second question, uh, a small question. I'm also handicapped, so will it be a barrier for my future studying in abroad? Like I'm asking because I don't know much about the country rules. For example, if they ask for dependent, sometimes when it comes to a handicap immigrant for higher studies, they usually have do they have rules or anything? So, but basically the question is like to do experimental research in science, in biology, chemistry, uh, for
All
a
right.
handicapped student. Yeah.
Okay. So I'll try to answer as quickly as I can, brief and concise. So any disability is there's no discrimination against any disabilities, and that will not be a deciding factor for a program to accept you. And that's not a deciding factor at all. And there is a huge amount of support for a student. For example, when the labs are designed, we always make sure if a student in a wheelchair is present, how can they access the instruments in the lab? How can they do the experiment? So the benches designed are it would allow you to do your work without any extra hindrance. So that would not affect you at all. And the other question was financial funding. I just want to mention real quick that if you are pursuing a PhD career, this is my experience, and this is the experience for almost every PhD student in the United States in the field of sciences. You do get a stipend for being a graduate teaching assistant or a graduate research assistant, and then you also get a tuition waiver, and that really depends on the department, but most departments do offer. Although the stipend might not be a whole lot, it's enough to get by. And so that reduces the financial burden and helps with the funding scenario. The reason why I mentioned funding was if you are able to acquire funding independently, then that is crucial for you to demonstrate that you are good at acquiring funding. So that's the reason I mentioned funding, and you don't have to acquire your own funding for your project because your lab would typically fund the work that you're focusing on.
The last question, like the question was like, what would you recommend to go for a PhD straight after your bachelor's? So to gain some experience and go in there, and if so, like what type of job opportunities you recommend?
So again, I want to say it really depends on what you want to do. And I know we have limited time, but if you see yourself as somebody who is assisting a lab, a master's would suffice. For example, if you want to be a lab manager in an industry setting, for example, or if you want to be an individual who's supporting the assays that the company is doing or the lab is performing, then a master's would suffice. But if you see yourself being a scientist in an industry setting, if you see yourself driving the research that you would like to pursue, that you like to drive. So if you want to be a lead, essentially, then you would require a PhD. Again, if you are unsure about the industry, maybe it would make sense to go into the workforce for a little bit and try that out and see if that's what you would like to do. I think if you work and if you come back, it still is fine. But if you go directly into a PhD program and then try industry later, that's fine too. But it really depends on if you want to be a lead in a lab or if you want to be more of a supportive role. So it really, again, comes down to, I know I said it a lot, it really comes down to what you want to do and what you would like to do.
Let's thank our speaker again, and Professor Samarakkody, if you don't mind, please drop your email address, any contact information in the chat box. So a
Okay,
lot
I
of
will do
students who, I mean, asked for your contacts to contact you for advice.
that.
Now I invite Dr. Induni Siriwardana, Secretary of SLAAS Section E2, to deliver the vote of thanks.
Yeah, thank you. Thank you, Dr. Samira. Thank you, Dr. Samira. So we have come to the end of a wonderful talk by Professor and Sanoji Samarakkody. I hope it was able to spark interest and enthusiasm in our young scientists, chemists to start their career as a chemist and not to stay or refrain in that particular path, but there are a lot of opportunities to deviate into many other chemistry related subject areas, and Professor Anne set as an example for that. So thank you very much, Professor Anne Samarakkody, for your valuable time and accepting our invitation and spending your time, and also answering a lot of questions by our young scientists or the young chemists. So thank you again for your wonderful contribution for this last event. And also, I should give a special thank to Dr. Samira Gunatilaka for coordinating and organizing this event and putting a wonderful webinar at the end. And also, I would like to thank Dr. Dinusha Uddukula, who is the president of Section E2 SLAAS, and all the committee members, and also everyone who has joined here as the audience, and to make this event a successful one. And last, I should thank the SLAAS Council as well. So always guiding and be there with us for the events that we do. So I hope this marks the end of a wonderful event. So thank you very much, everyone. Yeah, I will stop it.
So have a good day, Professor Sanoji, and have a good night to all the other participants. Thank you.
Thank you very much for having me. Have a good night, everyone.